Today my topic is mad cow disease. Bismillahirrahmanirrahim. Little by little, day by day, what is meant for you will find its way. Learning outlines of this presentation. In this presentation, we discuss introduction, epidemiological determinants, chain of transmission and communicability, clinical manifestations, level of prevention, and recent advances. By the end of this presentation, participants will be able to describe the epidemiological determinants of mad cow disease, clinical features, prevention strategies, and global updates. What is the mad cow disease? In cows, it is called bovine spongiform encephalopathy. In sheep, it is called scrapie spongiform encephalopathy. And when we talk in humans, mad cow disease is known as Creutzfeldt-Jakob disease in human, and transmissible spongiform encephalopathy is the family of the disease. Bovine spongiform encephalopathy, which is also known as mad cow disease, is a progressive neurological disease of cattle caused by prions, which are infectious agents made up of protein material. The prion proteins affect the brain structure of the infected animal, causes the animal to lose motor skills, and eventually causing death. Clinical signs include behavioral changes, coordination problem, weight loss, decreased milk production. There are two types of bovine bovine encephalopathy, classical and atypical. Neither form is contagious. Classical BSC spread through the ingestion of certain material, for example, brain or spine from the infected animal. The main classical BSC spread was through contaminated animal feed containing meat or bone meal from the infected cattle. The incubation period for classical BSC from the time of infection until the onset of clinical signs averaged 3 to 6 years. The first diagnosis of classical BSc occurred in 1986 in the United Kingdom, where most of the worldwide cases occurred. Since then, many other countries have confirmed the cases of classical BSc. If we see the picture from sheep to cow to humans, the scrapie is a fatal brain disease affecting the sheep and goat. First recognized in Europe more than 250 years ago, infected sheep will rub and scrape their skin raw and eventually die from the brain degeneration. In mad cow disease of bovine spongiform encephalopathy first appeared in 1984 in a cow in Britain that was thought to have eaten feed that included offal from sheep that harbored scrapie. Since then, authorities have found more than 178,000 cases of BSE. Creutzfeldt-Jakob disease, which is also a disease, a mad cow disease in human, is a human equivalent of the fatal brain disease. It is a rare disease, typical occurring in 1 in million older mature adults. A new variant, probably caused by eating infected beef, affects younger adults in the same way it attacks the brain of cattle. As we already discussed, what causes the disease? In all three species, the disease is caused by prions, which is infectious protein that are smaller than viruses. What makes prions unique among infectious disease agents is that they lack the genetic material. A prion exists in two forms. Non-normal proteins can change its shape to disease-causing form. The conversion of one prion protein into harmful form can trigger a chain reaction among other proteins. When enough bad protein forms, they create long fiber that destroy the brain tissues. Prions are remarkably durable. Standard procedures used in labs such as heat and chemicals are not effective in neutralizing the prions. If we see in the brain, there is spongiform appearance in the pathology. World prevalence, the first confirmed case in which an animal fell ill with disease confirmed in 1986 in United Kingdom. Subsequently, 177 people contracted in 2014 the disease and died of disease with similar neurological symptoms, known as new variant Creutzfeldt-Jakob disease. Three cases of variant Creutzfeldt-Jakob disease occurred in people who had lived in the visited the UK, one in each Republic of Ireland, Canada, and United States. Four cases were reported globally in 2007, and the condition is considered to be nearly eradicated. In the United Kingdom from 1986 to 2015, more than 184,000 cattle were diagnosed with peak of new cases occurring in 1993. You will see the graph. In 1997, the cases start increase, and 1991 it is a peak, and then gradually it declines in 2008. If we talk about the prevalence of this disease in Pakistan, actually the Creutzfeldt-Jakob disease is not well documented in this country, and therefore its prevalence in Pakistan cannot be assessed. There is only one research paper which describes the two cases of Creutzfeldt-Jakob disease from ongoing dementia registry in Pakistan, which is published in 2016, which present the first case of 62 years old female presented with myoclonus, and the second case was 72 years old female who was presented with generalized axial and limb rigidity, mutism, personality changes, and hallucination. Creutzfeldt-Jakob disease, which is also known as mad cow disease in human, is a rare degenerative brain disorder. It is also caused by abnormal proteins called prions that damage the brain cell. It is incurable and invariably fatal. Symptoms include rigidity, degenerative progressive dementia, anxiety, depression, memory loss, personality changes, and hallucination. Physical problems such as impaired speech, jerky movements. There are four types of Creutzfeldt-Jakob disease variant which acquired consumption of BSE infected neurological tissue. If cattle meat has come into contact with infected neurological tissue, it may harbor the prions. Average age of onset 27 years. Initially, generally gradual psychiatric and behavioral aberrations. Familial autosomal dominant mutation in the PRP gene, generally between codons 175 and 220, may depend on the specific mutation range 30 to 80 years of age. In this, there is neurological degeneration, depression, absent-mindedness, and fatigue. Sporadic, how acquired it is unknown, age of onset 65 years, and there is rapid neurological degeneration. Iatrogenic, recorded cases for neurosurgery with contaminated instruments, corneal grafting, transplant of exposure to human dura mater, implants infected with prions, and the use of infected human cadaveric growth hormone. Average age of onset any, and initial symptom depends on the routes of transmission. If transmitted directly into the brain, rapidly progressive neurological symptom produces a proceed ataxia, and growth hormone cases tend to be present first and primarily with progressive ataxia. Clinical feature: the time between the moment an animal gets infected with bovine spongiform encephalopathy gent and the onset of clinical sign is between 2 to 8 years. Therefore, clinical signs of bovine spongiform encephalopathies are found in adult animals, which may demonstrate some of the clinical signs: nervous or aggressive behavior, depression, hypersensitivity to sound and touch, twitching, tremors, abnormal posture, lack of coordination, difficulty in rising from laying position, weight loss, or decreased milk production. There are three phases of bovine spongiform encephalopathy in cattle. The first phase is low infectivity rate, and at this stage, cow does not pose a large threat to human and animal. Second phase, symptoms are not evident, but the infectivity level is very high. The prion agent is abundant in both the spinal cord and the brain, and the cow is risk to public health. Third phase include clinical symptoms, and then death follows shortly. Now, epidemiological determinants. Germ factor initially it was thought to have been caused by slow viruses. These are classified as slow viral disease. Now there is evidence that bovine spongiform encephalopathy is infectious disease believed to due to misfolded protein known as prions. Prions replicate by causing other abnormally folded protein of the same type to take on their misfolded shape, which then go onto the same, leading to exponential chain reaction. Eventually, the prions aggregate into an alpha helical beta pleated sheet, which is thought to be toxic to brain cells. The germ is not destroyed even if the beef or material containing it is cooked or heat treated. As we have prions are single molecule containing about 250 amino acids, they are abnormal variants of protein which normally occur in cell. They have ability to convert normal form that come into contact with their normal forms. If we see there, this is normal protein amino acid sequence in the sheet form. And when there is disease prions amino acid arrange in the beta helix or beta plated sheets, which is toxic to brain cells. If we talk about transmission can occur when healthy animals come in contact with tissue tainted with this disease. In the brain, the agent causes native cellular prion protein to deform into misfolded state, which then goes on to deform further prion protein in the exponential cascade. This results in protein aggregates, which then form dense plaque fibers. Brain cells begin to die off in massive numbers, eventually leading to microscopic appearance of holes in the brain. That is why this is called spongiform, degeneration of physical and mental abilities, and ultimately death. If we see in the picture, there is brain shrinkage and deterioration occurs rapidly. Brain section shows spongiform pathology characteristics of Creutzfeldt-Jakob disease. Transfer to how this disease is transferred to humans. Sheep is scrapie used in meat and bone meal known as offal. Infected beef eaten by human. Not affected by cooking, brain and spinal tissue. Maternal transmission is very low risk. Genetic susceptibility. Human have been homozygous for methionine at the codon 129 of PRPC gene. Possible modes transmission from the surgical instruments used for appendix of brain tissue, growth hormone injections, vaccines. Incubation period, bovine spongy encephalopathy is transmissible neurodegenerative fatal brain disease of cattle. The disease has long incubation period of 4 to 5 years and it is a fatal for cattle within the weeks to months of its onset. The nature of bovine spongiform encephalopathy then is still being debated. Levels of prevention. There's need the targeted surveillance of occurrence of clinical neurological disease, transparency in reporting findings of bovine spongiform encephalopathy, safeguards on importation of live ruminant species and their products, removal of specified risk material, brain, spinal cord during slaughter, and processing of carcasses and from the human food and animal feed chains, prohibition of inclusion of SRM in the animal feed, and thus removing potentially contaminated material from the food chain. Human destruction of all suspected animals exposed to prion contaminated feed. Banning the use of man meat in ruminant feed, further reinforced by mammalian to ruminant feed ban. Appropriate disposal of carcasses and all animal products. Livestock identification to enable effective surveillance and tracing of suspected livestock. Always vaccinate cattle properly. Disinfection of porous load autoclaving sodium hypochlorite is used, rendering at high temperature and pressure resistant in tissues and dried organic material hide tighter. Treatment diagnosis. Currently, there is no test to detect the disease in live animal or in human. Veterinary pathologists confirm BSE by postmortem microscopic examination of brain, medulla, spinal cord tissue, or by detection of the abnormal form of the prion proteins. These are some tests. All are based on antibodies to detect prion protein in tissue. Immunohistochemistry is considered to be gold standard. It is internationally recognized, expensive, and labor intensive. Rapid diagnostic tests include Western blotting or ELISA. Treatment. No treatment is available to slow down or stop the progression of mad cow disease or other prion infection. Studies are currently taking place to investigate a number of experimental treatments. Only treatment is prevention is only the available option. BSC risk status. Bovine, bovine spongiform encephalopathy is a disease for which the OE has established official recognition of sanitary risk status in countries in their entirely or in defined zone and compartments through a transparent and science-based and impartial procedure. The categorization risk status only apply to classical bovine spongiform encephalopathy. Atypical forms are excluded from the scope of the categorization because they are believed to occur spontaneously in all cattle population at very low rate. The OI officially categorized two categories of BSE risk status. The current requirement for recognition and maintenance of these categories can be summarized. Negligible risk, with regard to BSE agent for last 7 years, intensive active surveillance over 7 year period and maintained over time. No indigenous case of classical bovine spongy encephalopathy born less than 11 years old. Evidence of an effective ruminant-to-ruminant feed ban for at least 8 years. In control risk, at least control risk with regard to the bovine spongy encephalopathy agent, intensive active surveillance over 7 years period and maintained over time. A country's zonal compartment not recognized as having control or negligible BSE risk status fall into third category, which is known as undermined BSE risk status. Bovine spongian encephalopathy is a progressive fatal disease of nervous system of cattle. It is caused by accumulation of abnormal protein called prions in the nervous system. Two forms strains can be identified, classical or atypical. First is detected in 1986 in the implementation of appropriate control measures, resulting in the decline of classical BSE cases worldwide. It is considered zoonotic due to its assumed link with the emergence of variant Creutzfeldt-Jakob disease in humans. Bovine spongian encephalopathy is OI listed disease and it has established official recognition of sanitary risk status for its classical form. Bovine spongian encephalopathy surveillance USDA began conducting BSE surveillance testing in 1990 as a way to detect any case that might be present in the United States. If no cases are found, surveillance results also prove the absence of disease in the country. The current US surveillance program in place since 2006 far exceeds the current surveillance guidelines provided by OIE. The testing program focuses on cattle population that are at high risk for BSE, including animals 12 months of age and older that display central nervous system signs, and animals over 30 months of age that are excluded from slaughter due to poor health status. Producer can support USDA's effort to identify bovine spongy encephalopathy, especially cases of atypical that fall outside of normal surveillance effort. Immediately report any unusual or suspicious sign of disease to your veterinarian, state or federal animal disease control officials, or to your country agriculture agent. Watch cows that show nervous or aggressive behavior, difficulty with coordination, trouble standing up, decreased milk production, and weight loss. Report the location of any potential offspring for affected cows. These are rationale for surveillance, global surveillance of variant Creutzfeldt-Jakob disease and other forms should be lead to better understanding of disease, include potential causes of iatrogenic CJD and as well as distribution of various hereditary forms. Recommended type of surveillance. One center must be identified at central level to carry out surveillance. All designated, probable and possible causes must be notified by appropriate healthcare professionals. Check out death registration in order to identify cases not detected by routine surveillance. Recommended minimum data elements. Case-based record, subtype and classification of CJD, age, gender, country of birth, geographical information, occupation, date of onset and date of death, vital status, aggregated data reporting should be case-based, recommended data analysis, presentation and reports, number of cases by subtype, classification, geographical area, number of cases by the year of death and by age at death. Preference indicator of surveillance include time between onset of symptoms and reporting. In surveillance, as sporadic Creutzfeldt-Jakob disease in possible definition of possible case include progressive dementia, EEG atypical not carried out, and duration less than 2 years. At least two out of the following clinical features: myoclonus, visual or cerebellar disturbance, pyramidal and extrapyramidal dysfunction, akinetic mutism. Probable case include progressive dementia, at least two of the following four clinical features: myoclonus, visual or cerebellar disturbance, pyramidal or extrapyramidal dysfunctions, akinetic mutism with atypical EEG, whatever the clinical duration of disease, and positive 14 to 33 assay in CSF and clinical duration leading to death in less than 2 years. Definitive case include neuropathological confirmation and confirmation of protease resistance non-protein in immunohistochemistry or Western blot. These are the further definitions of surveillance of malleable CJD variant and variant case classification. Mad cow disease is actually the bovine spongiform encephalopathy, and mad cow brain disease appear to cause fatal human brain disease. Severity begins mad seizure months or years after injection. It can spread. How it spread? Person or animal eat food contaminated with brain or spinal cord tissue from infected animal. Disease attack the nervous system. Outer layer of the brain develop tiny holes, look spongy. That is why it is called bovine spongiform encephalopathy. Prevention: destroy infected form. Animals don't use animal products containing brain or central nervous system tissue as livestock feed. Now it is time for MCQs.
First time
First
is
MCQ
let's
is
talk
from
animal.
A.
yes, straight answer. Bovine progressive
Prion.
infection. A. Incubation period for classical KFC.
A.
First
First
confirmed case
confirmed
of
case
mad
of
cow.
mad cow.
B.
The quantitative event of mad cow disease is probably of Lenda cicatrium.
D.
It is not a symptom of mad cow disease in cows. Uncontrolled
Not uncontrolled. What types of
What types of tissue carry the greatest risk of
C.
some cases of human
some cases of human mad cow disease?
diagnosed
B.
what
What
must be
must be
done
done
to
to
destroy
destroy
the
the
infection of cow disease?
use of strong disinfection, avoid radiation, incubation and treatment. Insemination. It
It
is
is
not
not
considered
considered
irresponsible not to vaccine for personal contact.
Thank you very much, Dr. Khansa. Everyone, please mute your mic. Okay, thank you very much, Dr. Khansa. It is a wonderful presentation on a very difficult topic. Actually, this is one of the disease that we always have a very high index of the hardness that is associated with it, because we have three types of our important topics that have been mapped in any curriculum, that is according to their difficulty index. And some are very easy topics, as we have already covered, like measles, chickenpox. And there are certain topics that are hard topics, and even their diagnosis and their recent advances and their epidemics, outbreaks, and how to control them, how to prevent them. Again, all these come under hard or difficult index. The another important point is that you have made it so easy to understand and so comprehensible, and it is now becoming so easy and informative after your presentation that I am so glad about it. Today, as you all know, it was a foggy morning, and when I was traveling to King Edward Medical University, I couldn't even see the road properly. And at that time, I was have this in my mind that we have a class, and I have to rush and reach before time, and we have to read about a very important and have to discuss and present about a very important topic that is little bit difficult for the students. But when you have started and ended, it is such a pleasure to be with you all that it seems as if it is one of the easiest topic in our community medicine. So I have my first question from the audience. What is the another name for the mad cow disease?
Transmissible spongiform encephalopathy.
What is, please, I couldn't hear you. If you please a bit louder. If you please bit louder.
Spongiform encephalopathy.
Another, right? Very
Bovine
nice.
spongiform encephalopathy.
Bovine spongiform encephalopathy.
Very. I'm so glad that there are three people who are answering the same question. This is my experience as an examiner for so long that whenever I ask what is another disease of mad cow, they are looking as if I am looking at some cow. They are lost at some place in time. So in this session, I'm so happy that three students, they have come up with the correct answer. We also called it another name for it, Creutzfeldt-Jacobs disease, right? And this Creutzfeldt-Jacobs disease is a very important health issue, especially in those countries where they used cow as well as the other mammals for their primary source of proteins. Would you please tell me about the epidemics that have occurred in various countries? What are the top countries and how they have prevented that disease? Anybody from the class?
Ma'am, major epidemic was in United Kingdom.
Dr. Hansa, would you please answer this question?
Madam, first confirmed case in animals occurred in 1986 in the United Kingdom. Later on, 177 people in 2014 contracted the disease with similar neurological symptoms. And later on, various countries also reported cases, for example, in Ireland, Canada, and United States. But from 2017, four cases were reported, and the condition is considerably nearly eradicated.
Okay. Would you please let me know that why there are certain countries who are having this disease again and again? What are their deficiencies or why they are not implementing the preventive measures? What about the reasons that the prevention failed again and again, like UK?
And first of all, surveillance system is not, there is implementation gap. Uh, Health strategies present, but implementation poor. There is not monitoring and evaluation.
You have already presented about it, about the lab investigations and the diagnosis of the disease. And one of the bottleneck is that the lab tests and the diagnostics that are very expensive, and even they are not available at certain places. And even if they are available, their sensitivity and specificity is a great issue. So this is one of the main reason. Okay, right. I think you have covered it almost all the aspects very well. I have no other question to ask. Thank you very much, everyone. And if you have any question, you can ask. And okay, Allah Hafiz and best of luck. And I really appreciate your aptitude towards learning. And if you can come to the university in such a foggy morning where even we can't see the roads properly and we can't even see the our way to the university, and you all are here and you are learning, this is one of the great thing that I must appreciate. So Allah bless you all. Allah Hafiz.
Allah Hafiz.